Okay, good afternoon, everybody. Firstly, thank you for joining this webinar. Firstly, let me introduce myself. My name is Phil Souter. I'm a Senior Director at Procter & Gamble, working in our R&D department, looking at designing new molecules for our fabric and home care business. First of all, before we kick off today, I'd like to give a thank you to our sponsor, which is Cobotics, and give you a little bit of information about them. So Cobotics is a technology company developing the first self-driving lab for materials discovery, powered by AI and robotics. They're focused on accelerating the exploration, discovery, applications, and production of new molecules and materials. If anyone would like to find out more about Cobotics, their webpage is shown on the screen, so please feel free to visit that. So in terms of today, we would like to thank you for joining this webinar. It is the first in a series of 3 different webinars focused on digital design. So today, the focus is very much on digital design of molecules and formulations. We have 2 outstanding speakers for you. And then on the 8th of September, we'll be moving on to talk digital design of chemical processes. And that talk will focus more on manufacturing, including optimization of existing processes and also new process engineering. And then finally, on the 21st of October, we'll be looking to run a session on the digital design of life cycles. And this will focus on chemical tracing through the supply chain and a cradle-to-grave approach and the circular economy. So in terms of what you'll be seeing today, first of all, Caroline is going to kick us off with her talk. I'll introduce her in a second, and then Anthony will give a talk, and then we'll move into a questions and answers session. Now, I would like to say, if you could please submit questions throughout the webinar, we will then address them at the end in the Q&A session following both talks. Your Q&A coordinators are both on the line. That's Tom Harrison and Will Atkinson, both from SCI. And as we go through the talks, if you want to submit your questions, then they will collate them and make sure that we address as many as we can at the end. So our first speaker today is Dr. Caroline Lowe. Caroline is a computational drug design consultant who uses her skills in both medicinal and computational chemistry to create novel compounds with targeted biological activity. She currently works with global health R&D organizations, academics, and biotechs using a range of molecular modeling and cheminformatic techniques. She received her PhD from Imperial College and spent 20 years in the pharmaceutical industry, working with Sir James Black as part of Johnson & Johnson. Her work with the late Andy Vinter developed the molecular field point technology that's been successfully commercialized with Cresset. She's since moved back to Imperial in 2007 to establish computational chemistry in their Drug Discovery Centre, one of the first UK university centres to translate academic research into a portfolio of drug discovery projects. She has a long association with the SCI's Fine Chemicals Group and has organised many meetings on both computational and medicinal chemistry. So with that, I will hand over to Caroline and the talk she's going to give today on digital drug design, learnings and opportunities.
Right, thank you for the kind introduction and the opportunity to speak today about digital drug design. Some learnings from a project that was recently published, and some opportunities for you to hopefully use some of these techniques in your own research. Now, computer methods have a long history of use within the pharmaceutical industry. Everything from the point at which you find some interesting biology and you can conceive of a point at which you might be able to interact with human physiology to create a new therapeutic, through to engineering drug-like properties into a tool compound, to designing clinical trials, which is the point at which your compound will first be tested in man. Now, obviously, in a 25-minute talk, I'm not going to be able to cover the whole of this gamut of the whole of this area. So we're going to focus on some processes that we use to kickstart a drug discovery project. And in this case, this was a project to find inhibitors of a kinase called MAP4K4. This was an Imperial project and the principal investigator was Michael Schneider. And my role in this project was to produce the first chemical series that we could use as a small molecule to block the actions of this kinase enzyme. Michael had already done gene silencing in a human model and had spent many years looking at the processes which cause damage to heart tissue during heart attacks. And what he wanted to be able to do was to produce a pharmacological intervention, i.e. a drug, that could be used to give to people undergoing, when they're in the middle of a heart attack, to protect their hearts, the heart tissue. The project began in about 2012, and we were in a situation where we had no protein structure and no existing ligands known. So I'll take you through the story of how we managed to create some chemistry for this project. And in going back to this, I actually had a second look to see what sort of information there was available, and I will highlight some of the sources of information from the large chemical databases that exist, and illustrate how different approaches could be used today. If you want to read the paper, this is the reference. And when I'm showing pieces of software, I've also highlighted the web addresses that you can go to find out more about each package. Right, so before I start, here are the team. Here's Michael on the left-hand side, and Lorna Fiedler, who was his postdoc, who did much of the biology in this project. On the right-hand side, we have Sian Harding, who created the technology that allowed us to test the compounds in human cardiomyocytes derived from stem cells, in both 2D and 3D models. And in the center, we have the drug discovery team. We were all recruited with significant industry experience, and I've highlighted the three of us who were involved in this particular project. And I want to say a particular thank you to Katie Chapman here, who's on the left, who was not only involved in the discovery of these compounds, but also in their optimization when she moved to DomainX. So how do we go about finding these tools? Around about the turn of the millennium, pharma companies were building huge compound collections and carrying out robotic high throughput screens. These were wet screens. They involved screening very large numbers of compounds, say up to 1 million compounds. So you can see that this isn't a cheap option. Even if we reduce the costs of screening each compound down to $1 a well, we're still going to rapidly ramp up the costs of running a screen like this. And we now know that the chances of success from these types of screens is pretty low. 0.3% is accepted as an industry standard for this type of screen. So this led to the development of computational methods for virtual screening. Virtual screen is an entirely computer-based screen, and it's quicker and cheaper than doing the equivalent wet screen. The big advantage is that you don't need physical samples of the compounds, so you can screen huge compound collections. It's now becoming routine to screen compounds, collections of 1 billion, libraries of 1 billion compounds. Here, another alternative approach we could have taken would have been scaffold hopping. So to take a known compound and skip from one chemotype to another chemical series, whilst retaining the biological properties of that molecule. We might want to do this to improve the drug-like properties, the bioavailability of the compound, provide a backup series, or perhaps strengthen our patent position. This is an academic project, and we didn't have access to high throughput screening facilities, so we chose to take the virtual screening route. So what we're effectively doing is carrying out a series of design, make, test cycles through taking a biological hypothesis, finding a hit compound, translating it to a lead, optimizing that, and hopefully we'll come up with a drug candidate at the end. Although this is not always as simple as it looks, and sometimes requires several rounds of this cycle. So today I'm going to tell you about a virtual screening method that uses high content pharmacophores, a little bit about ADMET profiling, all connected to finding inhibitors of this kinase, MAP4K4. Now, MAP4K4 is one of 2,000, family of 2,000 enzymes. And this picture on this left-hand side is part of the tree of the human kinome. It's a pivotal, the kinase itself is a pivotal mediator of inflammation, cytoskeletal function, and apoptosis. And most of kinase inhibitors are tested for cancer, roles in cancer. But in this case, MAP4K4 inhibitors have been looked at for a number of indications. And in this case, we're going to look at it for heart disease. In terms of virtual screening, we have two options. The first are docking methods. These are structure-based methods, by which we mean protein structured. And the question that we're asking is, does this compound have a chance of fitting into this ligand binding site? So we had to rule out docking approaches at this point because we don't have any protein data. That leaves us with ligand-based methods. So here we're asking the question, can I find molecules with similar properties to a known ligand? And of course, it's the go-to method when you haven't got protein structures. Now, I should say that each of these methods are equally successful. You tend to see more pictures in talks of docking methods, but it's well known that both methods are equally efficacious. And in some cases, people use combinations of the two. So to set up a virtual screen, you're going to have to go through a process of generating or acquiring either a protein or ligand structures, preparing a database of compounds to screen. And these are, in fact, you can download lists of compounds from databases such as Zinc. This one contains 230 million commercial compounds and also a set of 100 million compounds which are pre-configured to be used directly in a docking experiment. You can find these at this website, Zinc Docking. And more recently, Enamine have launched their real space collection of 14 billion compounds. These are not commercially available compounds, but they're ones that should be synthesizable within one or two well-regulated steps. In running the virtual screen, we need to prepare the protein structure, assemble the ligand set, configure software, choose meaningful parameters for the calculation, have a means of manipulating the files. And this will give us a hit list of, say, probably several thousand compounds, which we're going to analyze. So we need software to handle that process. We're going to try and select molecules that have diverse chemistries. We need to be able to visualize results and have a means of purchasing the compounds and following up on those hits. Okay, so the plan for MAP4K4 was to use the fact that kinase inhibitors are rarely specific and to look for a small set of compounds which had good off-target effects activity at MAP4K4. And then we're going to use a virtual screen to identify novel compounds. So finding the first chemical starting point involved a wet screen. This was a compounds from the LOPAC set. It's a commercially available set of biologically active compounds of around 2,000 compounds, were tested against the kinase domain in a single shot cell-free screen, and any compounds that inhibited the activity of the protein by more than 80% were further characterized and filtered down to a set of 4 compounds fully characterized which had PIC50s of above 7. So this would be 0.1 micromolar. So we've got an overall hit rate of 0.2% here, very similar to what we would have expected from this kind of wet screen. Just to indicate how dirty these compounds, how promiscuous some of these compounds are, this is one of them, chempalone. Its structure is up here on the right-hand side. And this is the full extent of the kinome tree. Each one of these branches has a separate enzyme at the end of it. And I've colored ones which this compound has reported activity at. The size of the blob at the end tells you how active the compound was. And here is MAP4K4. And despite the fact that no activity has been reported against this enzyme or on any database, and the fact that the activity that we found was very similar to its primary reported activities at GSK3 beta and CDKs, a total of 37 kinase activities. other kinase activities are reported. This illustrates the fact that for this particular field, they actually have their own vocabulary for describing how promiscuous these ligands are. And on the next slide, you can see the other two compounds that came out of this, two of the other compounds that came out of the screen. On the left, we have a PD compound, Park-Davis compound, which has very high nanomolar affinity. And there's a second compound from Glaxo Wellcome with similar activity to chenpalone. There are no literature reports that any of these compounds inhibit MAP4K4, probably simply because nobody looked, and all show multiple activities at a wide range of other kinases. So there's clearly something about these three molecules that MAP4K4 recognizes. So what we did was to use a high content pharmacophore approach using Cresset software to define the field points, to define field points in the screener database of 1 million compounds with 100 field point patterns from each compound. Now, these field points summarize the electrostatic and van der Waals properties of the molecule, and what we get out of this screen is a hit list of commercially available compounds, which we can then go out and buy. And the next thing to do is to find a way of processing this data. So here are two packages. These are both open source softwares that you can download for free. On the left, we have KNIME, which is a very powerful package that I use routinely for data wrangling. And on the right, we have Data Warrior, which is the chemical spreadsheet package. KNIME is an object-oriented programming system. So you pick a module that does something. In this case, this one reads data from an Excel file. It then colors the cells, partitions the data, and creates a machine learning model. And you can then pick ones to visualize, plot out graphs, carry out some statistical analysis, and plot a scatter plot of the data at the end. This is a very powerful package, but it also has a great user community who involved in cheminformatics, who provide nodes specifically for doing drug discovery. This includes companies such as Vernalis and Lilly, organizations such as the EMBL, LASA, and contains the most up-to-date copies of functionality that is used on websites everywhere to handle chemical compounds such as RDKit. And on the right, we have a Data Warrior produced by Idorsia, a Swiss biotech. They developed this as their in-house software and released the front-end package, which contains quite a lot of functionality for public use. And we use this to collate structure-activity relationships, develop new hypotheses, share data, visualize it, and develop the multi-parameter optimizations that are so important for drug discovery. If you're interested in KNIME, I suggest you go and have a look on their blog. They've created a series of 8 interconnected web flows, and this pipeline is called Teach OpenCAD. Each one does a separate activity, so you can group your compounds by similarity. You can screen compounds using a machine learning method. You can plug it into a database and pull down protein structures or information about compounds. So we're going to process the data from our virtual screen. We have hit lists of 1,000 compounds, and these are ranked by the similarity of the similarity score between the template that we're using, pharmacophore template, and compounds in the database. First thing we're going to do is to remove any undesirable compounds which might be reactive, which are too large to be drugs, or so-called pains. Now, pains are compounds known as to interfere with biological assays. So they might be chromophores or fluorophores or redox active compounds. And there are lists of these compounds. So we can remove these compounds before we decide which compounds to buy. We further filter them down so that they had low molecular weights below the threshold of 500, and calculated log P's within a range of 0 to 4. At this point, I looked at the overlays manually and cluster compounds and picked out a set of diverse compounds, which were further filtered down to a set of 50, which we went out and bought. And then those 40 arrived. That 80% cutoff is about par for the course for going out and buying these compounds. And we were very pleased that 5 of them had PIC50 values of above 5. So that's a hit rate of 12.5% for this process. And here are the compounds that came out of the screen. This particular chemotype, there were 7 examples of this, and it was clear that modifying the substituents on this aromatic ring here would allow you to modulate their activity. So that's a good thing to look at, and it means it's a good compound to pursue. However, compound solubility was identified as a problem. Here I've uploaded this structure to a web service provided by the Swiss Bioinformatics Institute. It's called SwissADME, and it will calculate a variety of drug-like properties and plot them out on this these 6 axes. This is called a spider plot. If our compound was perfect, I would expect this red line to lie entirely within this pink area here, which has calculated values for lipophilicity, size, polarity, and flexibility of the molecule. And here you can see the value for the solubility, which lies outside and flags up this problem. So a medicinal chemistry campaign run at DomainX under a Wellcome grant produced this compound by adding on a solubilizing group to the same point on the aromatic ring. It also led to a 10-fold increase in activity. So we now have a compound with good activity, which is soluble and suitable for testing in further assays. Big question is, have we still got compounds which are completely promiscuous and hit every kinase known to man, or have we managed to create a selective series, which was the aim of the project? So here we have on the left and right hand side the results from 2 separate screens against, on the left, 141 kinases run at Dundee, and on the right, a larger screen of 376 kinases. Now on the left, this is a heat map, and red bars show you compounds which inhibit that particular kinase by 100%, whereas green bars show our positions, where the kinase activity is percentage inhibition is not, there's no activity of that particular kinase. So these are the template compounds, and you can see how dirty they are, how many kinases they hit, and how many red bars there are in this screen. On the 2 columns on the right-hand side are the first compound that came out of the virtual screening and the improved compound with the solubilizing group. And if we look at that molecule's properties in a bit more detail, we can see that the only significant activities are at kinases which are direct relatives, very close relatives of MAP4K4. They're on the same branch of the kinome tree, and the examples of MINC1 and TNIC. So we have managed to create a selective set of compounds for a kinase inhibitor. This is really quite a high level of selectivity. So fast forward to 2020. If we were going to go about looking at this project again and starting it again, can we find any other additional information that we could use in our search for new inhibitors? The first place I would look would be the Chembl database. This is a database maintained at the EBI. It's a curated database, which currently contains data from over 76,000 publications and 65 deposited data sets. And the database contains information about the structure of the compound and their bioactivities. So there are over 16 million bioactivities in this database. And 175 compounds are reported to inhibit MAP4K4. They range in activity between 3 pChembl values, which is equivalent to a PIC50 value of 3 and 8. So some of these are quite low, some of these are about the level of the compound, our final compounds. So this would give us options for further optimizing the compounds we already have, or perhaps carrying out some kind of scaffold hopping exercise. If I look on the protein data bank, which is the central repository for all protein structures published in journal articles, and some more actually, we can find that there are now 17 structures of MAP4K4, some of them of the APO enzyme itself, some of them just of the catalytic domain, and some of them with small molecules bound to them. In this case, this is a fragment. Gives you the name of the compound. It gives you an index, which you commonly find these structures referred to, and the structure of the compound. It also now has some quite sophisticated viewing software in it, which gets around a problem that I'm commonly asked, which is, how do I view these compounds and how can I see what interactions a ligand is making with the protein? It used to involve setting up some molecular modeling viewers, but now the software on the website itself is sufficiently good that you can just highlight your molecule, and here you can see there's a hydrogen bond between an exocyclic amine and a carbonyl group here, and a pi stack between the edge of this aromatic ring and a tyrosine residue here. So that gives us options to create to use docking screenings, structure-based design, or scaffold hopping. So in 2012, the published project that I described to you started from no protein or ligand information, but we were able to create selective inhibitors by assembling the off-target activities of existing kinases, and that series is now in development for cardioprotection against heart attacks. If I look at this, again, there's now an increased amount of information about this kinase, and so we have further options that we can use to create backup series. And as a final slide, if any of you are interested in finding out more about some of these software packages, there are a number of webinar recordings from Global Health Compound Design, series of Global Health Compound Design webinars. These are all available from the Medicines for Malaria Venture website and also via YouTube. And you can see talks from both the creators of NIME and the Warrior, if you want to know more about those packages, as well as a lot of drug discovery stories. Thank you for listening, and I'll be happy to answer any questions, I think at the end, and I'll hand back to Phil.
Thank you very much, Caroline, for that excellent talk. Thoroughly enjoyed it. And
thank you.
I think, as Caroline says, if you can please submit any questions as a reminder to online to Tom and Will from SCI. They're the Q&A coordinators, and they will assemble those questions, and we will have time for a Q&A at the end. But the second talk you're going to hear today is from Dr. Anthony Williams. Anthony joined the Center for Computational Toxicology and Exposure, which was previously the National Center for Computational Toxicology, in the Office of Research and Development at the US EPA in May 2015. Anthony is a cheminformatician and primarily focused on the delivery of the center's data to the scientific community via the CompTox Chemicals dashboard. His interests extend to the aggregation and curation of chemical data, the development of models to support physicochemical property prediction, and the development of software approaches to support non-targeted analysis. He has over 2 decades of experience in cheminformatics and chemical information management, with a focus on internet-based projects to deliver free access, community-based chemistry websites and services. Specifically, the ChemSpider database with over 80 million chemicals and 100,000 users per day. Anthony was previously the Chief Science Officer for Advanced Chemistry Development and VP for Strategic Development for the Royal Society of Chemistry. He has authored over 230 publications regarding computer-assisted analysis of data, cheminformatics, and chemical information management. So please join me in extending a warm welcome to Anthony, and I will hand over to Anthony for his talk.
Thank you very much. I appreciate the opportunity to present. We're all getting used to these interesting experiences online. So I'm speaking to you from Research Triangle Park in North Carolina. And I've been at the EPA for a few years now and building specifically a website that is responsible for sharing the data from our center. So the Center for Computational Toxicology and Exposure is working on coming up with algorithms and assembling data so that we can protect the public from chemicals that can be of concern. It's a very general website in some ways, so I'm hoping that what I show you, there are parts of it which will be of interest to you, and I will make sure that you have access to all of the relevant materials that I'm presenting here online so that you can download them. This is my contact information. Please feel free to follow up with me after the presentation should you have any questions also. Of course, we have the end of this session. There is a publication that's already out there. This is from the first release. This is open access, so feel free to grab a copy and you can understand then how we built it in some detail. The dashboard has now gone through 10 separate iterations. The last release was 2 weeks ago. At present, it has 882,000 substances, and we are pulling together all of these chemical substances and, where we can, physicochemical data, both experimental and predicted, hazard data, so we have data for over 50,000 chemicals. We have in vitro bioactivity screening data, so that we are pushing thousands of chemicals through thousands of bioactivity screens, and all of those data are made available. We also have built a link farm to tens of public resources. We're not trying to go it alone. We're trying to be part of the network of online resources that are available for the community to use. We built a number of integrated modules that allow for read across as well as literature searching against PubMed, and I will be showing you some of these modules today. And also we have, because we are capturing many tens of thousands of chemical structures, we're also able to build specific searches that support mass spectrometry, structure identification, because we do run a lot of data in our own laboratories where we are looking for chemicals in the environment. We also have, it's a single application integrating multiple resources, so the search capability is very flexible. I will be showing you a live demo at the end. We have all the tox data made available. We have the bioactivity data, similarity searches, so the basic cheminformatics searches are all built in. With the read across module I mentioned, PubMed search that I mentioned, as well as a batch search. So many websites will deal only with one chemical at a time for you to search. In this case, you can paste in a list of 5,000 CAS numbers, 5,000 names, and harvest data out in this batch search. I think this makes this tool fairly unique at this point. So the interface is very simple at the moment. It is text-based, so you can input CAS numbers, names, synonyms. We have millions of chemical names in here because of 882,000 substances. Some of these can have up to 300-400 synonyms each. But we're trying to deal with all of the complexity of pulling together all of these synonyms so that these searches will surface the chemicals of interest. Show you, we also have product use categories. So if you're interested in chemicals in mascara or diapers, things like this, then you can search on product use. And we also have assay gene searching because of the bioactivity work that we do. In this case, I'm searching on bisphenol. It's a type of search. This will surface all of the chemicals of interest that contain that term. And in this case, I'm going to select bisphenol A. Here we see the chemical structure. Plus you can see intrinsic properties such as formula, mass, monoisotopic mass. These are the properties that would be used for allowing us to do formula and mass searches to support analytical chemistry. And on the left-hand side, we see all of the various forms of data that are available for you to browse through. And I don't have time to show you all today, but there are videos online that at least an hour in length that will dive you into this in a lot more detail. The executive summary is a quick look at the type of information that we have available for the chemical at a toxicity level. So what we're mostly concerned with is the toxicity of these chemicals. So when you go to the executive summary tab, it will tell you what types of values are available. And as you browse through this, you can see that we tox data, chronic tox, developmental tox, etc. We take into account physical chemical properties, because this is specifically in regard to fate and transport and persistence in the environment. And we have regional screening levels that are of value for people who are considering the levels of these chemicals in the environment that should be of concern. And many other data that are all summarized in this one-page view. But this is a summary, so as you dive down, you can go into the hazard subtab on the left-hand side here, and you can find tens, if not hundreds, of values that are being extracted from dozens of websites that are online. We also pull data directly from literature articles, and we do our utmost to curate these data into the highest quality level that we can make available. It is an ongoing task with 882,000 substances, and we have phys-chem properties and hazard data, etc. It's an ongoing challenge to continue to curate these data to elevate it. We do have 5 full-time curators on staff working just on the chemicals, so making sure that the CAS numbers, names, structures are all mapped together. But we also have people working on each of the individual databases that underpin the application. So the hazard data specifically come from a database called ToxVal, developed by one of my colleagues, Richard Judson. Over 800,000 toxicity values, over 50,000 chemicals, 30 sources of data, and 70,000 literature citations, including an integration to other resources built at the EPA, specifically the ecotox data. When you dive into the properties, you can find both experimental data, and in this case, we can see just a short list of the phys-chem properties. We can see we have log P or log K or W, depending on whether you work in environmental sciences or drug discovery. We can see we have one value here based on the number in the parentheses, 7 melting points. And when we don't have property data, it's not every chemical structure that we have or chemical substance is going to have property data that's available experimentally. When we have chemical structures, we can predict these properties using QSAR, so quantitative structure activity relationships, or QSPR is another term. So we have assembled large curated data sets of log P, melting points, water solubility across the board, and we've been able to build these open source prediction algorithms. Our set is called the Opera predictions. You can download the prediction suite offline, so you can run in your own environment, and we will predict these properties for the chemicals that we have. So in this case, while we have built our own predictions, there are many available, including systems that are available from commercial vendors such as ACD Labs. We have the EPI Suite set that was built internally under contract for EPA. It's been around for 20 years. So we can show you many different predictions from different sources. But for our own, we also give you more transparency. So you can see here a hyperlinked report that allows you to see there's 14,500 log P values, the distribution of those log P values. You can see the absolute details of the performance of the algorithm. So for the 882,000 chemicals, about 800,000 of them have 13 different endpoints predicted. So we're talking over 10 million calculations plus all of these detailed reports, which include showing you the nearest neighbors that are being used in the training sets to produce these outputs. So a lot of transparency about how we do modeling. So we will give you all the training sets, test sets, etc. Total transparency plus open source models. This is a view, just a summary view of the bioactivity for bisphenol A measured against hundreds and hundreds of bioactivity assays. All of these data are available for download. For a particular chemical like this, you can download the data into an Excel file and work with it offline. And not only have we got the summary view, but we can actually show you the dose response curves for each of these specific measurements. So in this case, there are 136 of the 850 assays through which bisphenol A has been studied have active hit calls, and you can see the individual dose response curves. So again, a lot of transparency into the data. We're trying to make everything available in consumable form And should you wish to take the data offline for your own systems, you can do that. You can download it, put it in your own databases. This may be of more interest to this particular group, and that is the fact that because we're extracting chemicals directly from safety data sheets from various sources, for example, Home Depot, Walmart, etc., we have systems now where we can do text mining across these documents, extract the CAS numbers, the names, the weight fraction when they're available. We can blend all of this into a single database now to produce you access to finding, again, as I mentioned at the beginning, find all chemicals in diapers, find all chemicals in mascara, in shampoo, in hair dyes, things like this. So this is a view of where you would find bisphenol A. And you can find, as I said, the chemical weight fraction. We even have exposure predictions as a function of age, sex, body mass index, etc., because we've been building these algorithms over many years. I mentioned the multiple synonyms. This is a display of the synonyms that we have available for bisphenol A, just a small subset. I believe we have well over 100 for bisphenol A. Aspirin has 300, something like this. The reason we put this all together is because wherever you are, you may, in the world or whatever the article is that you're reading, you may come across a synonym that needs to be mapped in order for you to find this particular chemical. So there's many systematic names, multiple languages, multiple trade names, etc., etc. This will also allow you access to things such as the names of herbicides and pesticides that are in the marketplace that you would find on a shelf in a store. When you search on those particular names, then you can access the associated chemicals. So I showed single chemicals. We of course have multiple views here. So I searched on bisphenol A. Now what I'm doing is I'm going back to that search page and I'm just checking the little box that says search for this specific substrate. And what I'm looking for is a set of dyes that have color index codes. So searching on CI, I've turned up 1186 chemicals that have CI in the synonym. So this is a set of dyes. This is the latest release version, so we're approaching 1200 CI dyes, and we are adding chemicals literally daily. We have 5 full-time curators. We have a specific project going on right now to look at azo dyes, and we've added a few more hundred since then. So in the next release, we'll probably be crossing a couple of thousand dyes. Not just chemical structures, however, because so many of the chemicals that we are concerned with that are in commerce are what we call UVCB chemicals. So these are unspecified in terms of chemical structure. They're variable, so they may be C4 to C12 surfactants, linear and branched. They can be very complex mixtures. So these are also biologicals, so corn oil, nutmeg oil, things like this. So in this case, what I've done is I've searched on the synonym resin to determine all of the chemicals that we have inside the application that contain the term resin. We've turned up 1,518 chemicals here. This is specifically of interest to a project we're working on right now to try and determine how an application like this can be applied to these complex chemicals that are way more complex than simple chemical structures. Single chemical structures are hard enough. Complex UVCB chemicals are very, very challenging, especially in terms of integrating data. As I mentioned, we have full-time curators checking the data. So in this case, we show you for every record what level of curation chemical has gone through. We have 5 levels of public curated data, plus we have 2 levels that we retain internally. So we have an additional 18,000 chemical substances internally that we have not released yet, until we put it through a level of curation, which will elevate it to these 5 levels. These built-in modules include a module called Abstract Sifter. So in this case, what we're doing is we're integrating the application to PubMed so that there are 30 million abstracts to search. And what I'm interested in is a particular chemical. In this case, I've chosen morphine, and I select a query term. And when I select this term from this drop-down menu, then what it does is it adds a specific query into this box and associated with the CAS number and the name. There are mappings behind PubMed that will allow for these the multiple chemical names and CAS numbers to be mapped to this small entry subset. But when we add this query term, then we can hit PubMed in real time using web services and retrieve data. So if I was after water and exposure, as you can see, it's appended this specific query to the end of this box. We hit submit, and literally in less than a second, it's going to search 30 million abstracts. In this case, there were only 37 articles loaded. And then what we start to do is we start to filter through here using terms. So I want to find everything out of those 37 abstracts that were downloaded. I want to find everything that says wastewater, spectrometry, and EPA. So I'm looking for methods measured by EPA on mass spectrometry in wastewater for morphine. That's pretty much the approach. And what it does a frequency count of those terms in the abstract and shows you those counts assembled. And I can look at the relevant articles by clicking on the title. It will show me that the abstract with highlighted terms, and click to go directly into PubMed. There is a version for this that is available in Excel, so you can use it at your own desktop, and you can not hit it with a single chemical, but 100 at a time. So I recommend people take a look at Abstract Sifter. We also have mapped relationships in here. So for bisphenol A, for example, there can be multiple salts. BPA could be part of a mixture component. We take this approach because when we're detecting chemicals in the environment, we'd like to consider all of the potential sources by which that chemical found its way into the environment. So salt forms, multi-components, etc. And we do all of this in the background using cheminformatics mappings so that you don't have to try and harvest the data with small queries and long-term searches, so they can take a lot of time to try and disentangle all of this. We've made it one click. So if you want to see all of the chemical forms of bisphenol A across 882,000, one click will show you mixtures, components, and neutralized forms. And what we've been doing here is we've been mapping together all of the chemicals by distilling things down to what we call an MS-ready form. So by mass spectrometry, you're not going to distinguish all of the various salt forms and all of the various stereo forms. You're going to detect a unique chemical, so in this case, nicotine, but it will map, algorithmically will map to the various stereo forms. So we see the two D and L's nicotine here, the salt form, the isotopically labeled form, as well as the multi-component. So this is all done. So for any chemical that you searched in the application now, we will show you all of the substances in the application that contain that chemical. This is an example for BPA where we will take BPA, click on that search, and we find 27 individual chemicals where we have the barium salt, the sodium salt, we have multi-component systems, and we also have the 5 isotopically labeled variants of BPA that are hidden. From here, we can download this particular data set to an Excel file containing all the data. We also have multiple chemical lists and categories with 882,000 chemicals. We have done this by assembling over 300 individual lists. We surface about 280 of them at this point for the community to use. So if you're interested in finding categories of chemicals, you would come through and find all polycyclic aromatic hydrocarbons. I showed you could do a search on CI for color index levels. But when you do these searches, you would like to pull these data in a manner that you can download, repurpose, reuse. And I will be showing you this live. So for example, you want to go into the list of chemicals and find chemicals identified in human blood, plasma, and serum. Always under constant curation, more chemicals are always being detected. But one click and you can access this entire list, the 927, it's limited list out of the 882,000. I will show you many other lists. In product use and categories, as I mentioned, in this case, I'm searching for lubricant. And you can see there are various types of lubricants here. So home maintenance lubricant, auto lubricant. When you choose this particular category of interest, then it will show you the chemicals that have been detected in lubricant as isolated from safety data sheets. And there's over 1,700 of these chemicals associated with lubricant. And as I mentioned, lots of UVCBs in commerce. So some are structures, some of these complex chemicals. And so we see here soy oil, fatty acids, tall oil, etc., etc. Batch searching, while you may be interested in a single chemical or a single product use, such as lubricant, many people are looking for bulk data on a set of chemicals. So, for example, you might have a specific formula that you would enter in and find everything that matches that formula. That's an analytical science approach. Same thing for mass and error. What about if you were searching for 1,000 names that have been identified in a set of literature articles, etc.? Then once you've done that search, you don't want to stick on the application. You actually want to download the data into a form that you can use, and that would be an Excel file or a SCI file. And maybe you want to add all of the predicted properties. Check a box to find out whether or not there is hazard data available. Have we looked at through at bioactivity screens? How many consumer products are those chemicals in? So that's what the batch search is for. So as an example, sitting in a presentation at PitCon in Philadelphia a couple of years ago, a lady was giving a talk on opioids. She was saying how long it had taken them to assemble the list of chemicals for this review that were opioids. So what I did was I went online, pulled the PDF file. It took me longer to pull the names out of the PDF file because PDFs can be quite challenging to extract things. Copy paste can not work all the time. So I pulled out the list of chemicals. I pasted the list into the batch search and then clicked one button to pull down all of the names. We had every one of these opioids was in here. These are unique substance IDs. These are hyperlinked out of the Excel file. And then I wanted to add more information. So not only did I want to grab whether or not these chemicals were available, I also wanted the CAS numbers, the formula, the masses, the smiles, toxicity data, etc., etc. So the entire process is to check some boxes, click download, and then it will produce a file of interest. So this application is an integrated hub for environmental chemistry, primarily to serve computational toxicology. However, if you're not a computational toxicologist, the benefits are still there. So we have done an incredible amount of work to curate data in the public domain, full-time curation, to ensure that we're getting the right data mapped together. All of the access to the data and the models is fully transparent. Many of our models are open source. You can download them and use them. And we expand the data on a daily basis. And this data quality is key for us. We want to make sure that we have the right mappings between chemicals, toxicity, it's an enormous team of people that have been building this. I'm one cog in a very complex set of gears, and we've worked with lots of open public domain resources to make this happen. So I'm now going to go directly into the application. This is available to everybody in the world. You don't need to register. You don't need to log in. You can simply go to comptox.epa.gov. If you go to that particular URL, you will land at this specific page. We have numerous applications that we've run over the years, that, and we're now assembling them all into one single application that I'm speaking about. I'm now going to go to the Comptox Chemicals dashboard, and we're going to do a search. So if I search for something like atrazine, I'm going to go into this particular chemical structure, and we're going to learn what we have about this particular chemical. So this is going to be whether or not we have properties, whether or not we have hazard data, whether or not we have screened the chemical in multiple screens. It's going to be a little slower because I'm running video. So here's the chemical structure. I want to find all chemicals that have a matching formula. Click one button and it's found 7 related chemicals. If I want to see whether or not we have properties, I can go into the properties, and we have 1 log p-value, 6 melting points, 5 water solubilities. If I want to see these data, I can see what literature articles the data came from. So it tells you exactly what those literature articles are. And if I want to see the predicted solubility, then I can click here. And as I mentioned, we have this detailed report showing you the appropriate data that have been used, training test set, as well as multiple chemical chemicals that we have in the training set, and we have multiple values, experimental values for atrazine directly in the training set. So that would be looking at properties, and we have many properties. And when we don't have experimental data, we predict those values for you. We want to look for hazard data now. We can look at various types of data. So points of departure data, we have 444 records where we have information such as repeat dose, chronic toxicity, subchronic, the species that those data were measured in. You can see all of the data points here. So this is just 100 of the 444 data points. And if you want to download all of these data, as I've said, we want to make this as accessible as possible to you. One button click and it will download these data into an Excel file. And this is now in a consumable form that you can use directly for yourself. So this is going to load it and just display the data in Excel. Every time you see a table on the application, it will be available for you to download and view in this form. We've also recently done integration to safety data. So this is all of GHS data. They're available from various sources around the world. In terms of exposure, we want to look at what atrazine could be used for. We've recently expanded our resource, so we've got 558,000 consumer products that have been we've extracted data from. So if you want to look for individual data types, so for example, maybe active ingredients in pesticides, this is a category of data that we can have. So you can dive down into this. And depending on what your interests are, you might want to look for printer inks or toothpaste, etc., etc. And this is now downloading the 4,537 chemicals onto the desktop. Multiple types of information available, including integration to other resources from the EPA, such as the Toxics Release Inventory, which is going to show you the mapping of where atrazine has been detected in the US at various sites, etc. I mentioned the similar compounds. So in this case, we are simply showing you chemicals that are similar based on Cheminformatics Tanimoto search. Related substances would be things such as transformation products. So you could find chemical degrades in the body as a metabolite, or whether it degrades in the environment. I mentioned synonyms. We can see here we have many synonyms, and there's over 102 synonyms for atrazine. Many of these will be commercial products. I talked about literature, and we have a Google Scholar integration, but I find PubMed to be more informative in many cases because it's more of a focused search. And I want to know about atrazine as a hazard. Don't forget, we're searching 30 million abstracts, and it just did that. It retrieved 29 articles in real time. They're now at the desktop, and I can do specific searches. And it's doing a count analysis, and I want to look at this particular article, and it will show you the highlighted terms directly inside the application. And then if I want to read that article, one click and we go into PubMed. So now we've given you an easy way to navigate PubMed through particular chemicals of interest. Very quickly, I want to show you the list. So I mentioned we had dozens and dozens of lists. We are approaching, I believe, about 280 public lists, and these can be of different types. So, sorry, 267. So you might be interested in pesticides. So do a search, and it will take you into the 4,000 chemicals that are deemed to be pesticides according to the EPA's chemical search list. You might be interested in drugs of different types. So in this case, we've got antibiotics, we've got opioids, statins, etc., etc. So these lists are assembled to serve specific projects and capabilities that we need. The batch search, this is where you would input a set of chemical names, for example. So we can put toluene, bisphenol A, CAS numbers. We can mix these, and you could put in up to 5,000 identifiers, and then display those chemicals. And from here, we can download all of the data that we might have interest in. If we don't have a chemical of interest to you, for example, you do a search for a specific chemical based on cast number and name, and we don't have it, you can always do a search here on a chemical, modify this chemical to do real-time predictions. So as an example, we could do this, or just start from a blank canvas and draw a chemical of interest. And now what we're going to do is we're going to predict these properties in real time. So this is QSAR in real time. So this chemical is now being sent to a server. The descriptors have been generated. It is run through 4 individual models of different types. Each model will give you a slightly different result, and we've given you a consensus result. This all happened in real time, as I said, and it will show you the detailed calculation report of how the calculation was performed. The last part I would like to show you is I'm going to download a list of chemicals to the desktop just to show you the power of batch search at this point, so that if you have a set of chemicals and you want to harvest data from our application, how would you do that? So this is the acute exposure guidelines. This is a set of 174 chemicals. I'm going to download that. Every list can be downloaded to the desktop. If you're a cheminformatician, if you work in the digital world in terms of software and you're doing drug modeling, et cetera, you might want to be able to download in an SD file format. But in this case, I pulled it down to an Excel file format. What I'm now going to do is I'm going to take some of the cast numbers, some of the names, and we're going to do the searches. So in this case, I'm going to take a list of cast numbers into the batch search. And I'm going to search on both casts and names. So here's a list of cast numbers, and here's a list of names. So we're searching for both at the same time. And I might even be interested in, if you understand what an INCHI key is, we want to take a set of INCHI keys also. So these are very commonly available in your informatics world today. So I'm going to search INCHI keys at the same time. So there's 3 forms of identifiers all put in. And what I want to do is I want to display the chemicals to see if how many of them we might have. So now this is doing that search and finding me that list of chemicals. And I'm going to send them back to the batch search. Now I want to download the data. And this is where you can harvest everything that we have. Cast numbers, the INCHI keys, the UPAC name, the smiles, the formula, the mass, whether or not we have toxicity data, whether we've screened them, how many PubMed articles, and how many consumer products contain those chemicals. So for those data, one button click. The default is Excel, but you could also do this into an SD file, tab separated file. And this will be the last part of the talk. I'll probably reach my time, but hopefully you can see the power of being able to use an application like this, where to take advantage of the agency's efforts to pull together data and produce an application where you can access these data with just a few clicks. And here we have that data set. Here's the names, here's the cast numbers, the INCHI keys, smiles, formula, mass. Everywhere you see a Y, we have toxicity data. This is the number of consumer products containing the chemical. So I'm going to stop there, and I know that the questions will be asked from this point forward, and I will hand back and hope this was of interest. Thank you.
Yeah, thank you very much, Anthony, for that excellent talk as well. So yeah, just before we pop into the Q&As, just a couple of quick reminders. Yeah, we did cover this at the beginning, but we have 2 more in this series of SCI webinars that are supported by the Chemistry Council Innovation Committee, which basically is a joint government industry body that focuses on the future of UK chemistry. And one of the areas it's particularly interested in is AI, data, and digital. And these 3 talks are part of our work to support that. There's another couple of upcoming webinars shown on the bottom there if anyone is interested in learning a bit more about PPE that should be coming up soon as well. Beyond that, if you're interested in the SCI Talk series, then again, here are a few more dates for your diary. So upcoming lectures that you should be able to attend as well. So with that, we will now move into the question and answers. So Tom and Will, as the Q&A coordinators, want to give every opportunity to people to have their questions answered. So if you wouldn't mind sharing the first question.
Hi, Phil. Yeah, so got a first question here for Anthony from Samuel Meredith, who asks, regarding CompTox chemistry, do you have any plans to integrate information relating to polymeric materials, such as microplastics and bioplastics, into the database, including information about accumulation, residence, lifetime, etc.? And he says it looks like a brilliant resource for data-driven chemistry. Thanks for the talk.
Thank you for the positive comments. The answer is yes. We already have polymers in the application, tens of thousands of them actually. Those would be classed under IU, VCB chemicals. We are presently working on a resource called Nanobase, and that is spelled N-A-K-N-O-W-B-A-S-E, Nanobase. If you searched on Nanobase on Google, you should find links to some talks that are being given. It is a nanomaterial database specifically, and we already have a set of nanomaterials in the application. That's an example of a very focused study effort where the types of information that you're talking about is being curated out of other databases as well as literature articles. All of these data ultimately will end up in the application. We have to do the research first, and then we push it out in later releases. So residence time, for example, how polymers break down in the environment. This information will all surface eventually. So we do have efforts. Not all of the data that we are performing research on at the moment is yet available. The application is always behind the research that we're doing.
Okay. Do we have another question, Tom and Will?
Yeah, I have a question here for Anthony by Bruce Duncan. It's a question in two parts. So first of all, are house dust results included in the exposure sources? Are there any plans looking ahead to handle this data? And also, is it true that from the abstract Sifter that you can obtain a list and then use CompTox to rank by hazard and then key in on products that might benefit from product substitution? So I think there's two parts to that question, but if you can, yeah.
Let me start with the first one and then repeat the second one. I think I got it. So house dust, if you go to our list of chemicals, so we have a non-targeted analysis group that work inside the agency. We work very closely with them. A lot of the work we've done on the analytical science side, so formula searching, mass searching, the development of this approach to standardized chemicals, the MS-ready approach, where I showed you how we distill the chemicals so they all relate. And that was all done to support non-targeted analysis. If you look on Google, perform a search for non-targeted analysis CompTox, it will surface probably a dozen articles related to how the application can be used to support that. There are a couple out there on house dust, and how the application has been applied directly to the analysis of chemicals in house dust. We have run a project called NTACT, which is the EPA, ENTACT, EPA Non-Targeted Analysis Collaborative Trial. So it's over 30 laboratories around the world where we have provided physical samples of over 430 chemicals, including mixtures. Some of those mixture vials have up to 200 chemicals in. And the individual labs are trying to figure out how to detect each of those chemicals and break it out. The reason I mention that is because a lot of this work is directly related to the house dust analysis. When you look at these complex mixtures, you need to figure out what is in them. on the lists of chemicals on the application, and I showed you at the end there how to go into lists of chemicals, we have lists of chemicals previously detected in house dust and reported from literature articles and data sets. And there is a standard sample from NIST called, I think it's SRM 285, which is the standard house dust sample. That list is on there, and we have published on our own analysis of that. When you have that chemicals, you push it into batch. Now you can harvest everything you would want: toxicity data, hazard data, exposure level, predicted exposure levels, etc., etc. I covered question 1. I've forgotten question 2. So could you?
Yeah. So the second part was so the second part says that presumably from the abstract sifter, one can obtain a list and then use CompTox to rank by hazard, toxicity, and key in on products that might benefit from product substitution. That's the question. Is that true?
Yes. Okay. So abstract sifter at the moment, its existing implementation is single chemical. So at the moment, if you come to the application, you come in at a chemical level, and then you go into abstract sifter. So you end up looking for that CAS number, that name, and its multiple synonyms in PubMed. So it's single chemical at this point. However, the version that you can download into Excel, it's an Excel add-on. There you can work with multiple chemicals, and you can, when you have a list of chemicals of interest, then you could go back to the batch search, paste that list of identifiers in, go back to the batch search, then you can harvest predicted exposure levels for the community using tools such as Scene3 and ExpoCast. Those are our predictive models. You can detect, today you can say whether or not we have hazard data, but we don't provide the export of that data in batch search in the manner that I think you would want. But we can certainly tell you whether we have the availability of data. You can use the bioactivity screening and the counts, the number of active hit calls against multiple screens of different types, to help indicate whether or not a chemical has greater bioactivity. And you can harvest the number of consumer products. So you now have a list of chemicals that you might want to be concerned with, and you could rank order those and say it's this top list of 5 chemicals that we now want to find alternatives for. You want to find whether or not there are other chemicals that relate and use a read across approach. So what you would do is the genera module. So this general read across, I did not show it to you today. That is built into the application. You can take a chemical, look at all of its related chemicals based on toxicity at an organ level, organ specific level, and figure out what related chemicals you might want to consider for replacement. I hope that answered it.
Excellent. Well, thank you. Tom, another question perhaps?
Yeah, thanks, Anthony. So I've got a question for Caroline now. If virtual screening methods are both cheaper and often more effective, why might a project still choose to use a wet high throughput screening method?
Virtual screening is great, but you're never going to get away from the fact that you do have to do some wet screening at some point. So from my point of view, the real benefit of it is that you don't have to screen so many compounds. And I think that's its primary feature.
Excellent. Okay, that was a quick fire response. I've got another question for Caroline there in that case. Yep. Before we move on, so Blessing asks. She says she's a recent college graduate and wants to go into medicinal chemistry with an interest in computer-aided drug design, and is looking for things that they can learn, free software, etc., that will get her acquainted with the subject. So, could you provide any suggestions?
Well, a lot of the packages that I described in my talk were all open source. In fact, I think everything apart from the Cresset screen you can get your hands on just by downloading them directly from those websites. If you want to know how to use them, for example, Data Warrior has a limited online manual, but the creators of Data Warrior have done at least, I think, 3 webinars for the Gates Foundation for the global health community that you can also see for free. So there's a lot of free information out there. You just got to go and have a look at it and take your time, and you'll be able to start using these pieces of software. So for example, if you look on the 9 website, there are some nice tutorials. I pointed to those 8 workflows that they've put up there. Those are written by industry experts, and you can use them as they are, or change them to configure them to look at your own data. So you can try and see what happens with your data or a set that you've just downloaded from a large online database like Campbell, and have a play and see how you get on.
Did you have another question there, Tom or Will, or shall I?
Yeah. I've got another one for Carolyn. So you mentioned that you remove pains candidates prior to obtaining the materials for experiments.
Yeah.
Do you believe this means we're misimporting candidates? And could the modeling be used to get around these possible misses in experimental work?
Actually, what I do nowadays is not to remove the pains, but just to flag them. So if they come out at the end, I know it's a pains, because there are, as the question is probably alluding to, there are existing drugs out there which would have been caught by pains filters. So you would be missing out on some compounds if you took them out. So my practice now is to just flag them. So I know there's a problem there, and there might be something that I want to change about a molecule when I'm going through the optimization process.
Excellent, thank you. There's one more question here for Anthony. I suppose this might be one of the last ones. So from Simeon here, who asks, I see that there is a way to batch search for compounds related to certain information, such as formula and entry key, but is there a way to find all compounds that have experimental values for a specific property, such as boiling point?
Today, no, but you can go to the downloads page, which is the last item on the header, and you can download from there our experimental data dumps. So we give away all of our log P data, water solubility, melting points, boiling points, etc. So they are available. Those files get updated generally within a month of us doing a release. So the latest and greatest is not there, but we added 7,000 chemicals in six months. So the largest collections, 875,000 substances, you'll be able to download those data from the downloads page. We do intend to add to the advanced search, which I didn't show you. The advanced search gives you formula mass searching. We do intend to add the ability to search on all data types. So you want to find all boiling points, all melting points, etc. That's future plans.
Okay, great. Well, maybe I can ask one of my own then, which is to you, Anthony. I mean, in terms of, as you think about structuring your databases on polymers, whilst we've got the sort of SMILES methodology for small molecules, how do you think about structurally tagging the polymers?
Yeah, that is very tricky. So one of the ways we have done this is if we have repeat units, we use a technology called Marcouche, which is a Marcouche representation, allows you to repeat units in at least. So that's a very, very manual process. There's no easy way to do that. It literally requires that a curator sits down and hand draws each individual monomeric unit and map it as a Marcouche. So that's one path in. What we also do is I mentioned something called related substances, and I showed for atrazine, I showed that we have environmental degradants, metabolites, etc. For polymers, what we will do is we will map the individual monomers to the polymers. So those individual small molecule structures can map to the polymer. By doing that, it gives you the ability to come and search for a monomeric unit and then see the mapped polymers. Again, a very, very manual intensive approach when you have 882,000 substances to deal with. But we are making progress on it. And every release, we get more of those related relationships in space.
Excellent. Well, thank you. Right. So before I close, Tom and Will, are there any final questions?
Yeah, there's one last question for Anthony. So the the CompTox database, does it have data on the removal of compounds from water or physical properties such as viscosity, thermal conductivity, or explosive limits?
So we do have some predicted properties. I believe viscosity is in there. Conductivity, I think, was mentioned. I don't believe we have that. We do have lots of information about chemicals detected in water. And then from there, you can actually use the abstract sifter to migrate out to articles of interest. I can't remember the actual detailed question. Was degradation of chemicals in water? Was that what it was? You repeat that?
On removal of compounds from water. So, for example, yeah.
Not directly in the database, but you could certainly, come in at a chemical level and then use abstract sifter to look for that specific set of articles. This is the 10th release in 5 years. We are presently re-architecting the entire application. So the next version will be released in March 2021 with more capabilities. And if anybody from this call would like to follow up with me and make suggestions about what they think could be of value, we gather this information, and if a lot of interest is shown, more votes influences some of our activity. I am interested in getting community input on that.
Excellent. Well, actually, I think that last comment sort of sums up a lot of what we've seen today. I mean, I do want to thank both speakers again. I think, two excellent talks, and, hopefully you've all been very impressed by the amount of open source information, that one speaker's creating and the other speaker has been using. And I think it's a good testament to the transparency of science, and to both speakers. So thank you very much for being so open and transparent on the work that you have done. Hopefully this will be an inspiration to a new generation of chemists who are coming along to get themselves digitally acquainted and start using some of these tools. So yeah, in closing, thank you very much once again to the speakers, the delegates, and with that, we would like to. Sorry, go on, Caroline.
Thank you for the invitation.
Oh,
enjoyed
no,
it.
not at all. And thanks both for giving up your time for this. We really do appreciate it. And with that, we'll look to close this webinar. But thank you very much for attending, and we hope to see you at some future ones as well.